In our final lecture on muscle, we're going to look at what changes take place in muscle in response to various types of exercise. When muscles are activated during exercise, we need to think of some of the changes that are taking place, how they're detected, and what might be some of the consequences that mediate these adaptations to exercise. So if we think about the various stimuli that might activate pathways that lead to muscle adaptation, there are a number of obvious candidates. A number of times we've spoken about calcium. Calcium is fundamental to muscle contraction, and it activates a number of processes within the muscle cell. Changes in energy status. As the muscle breaks down ATP and other important fuels, they are potentially signals that could lead to adaptation. Changes in the redox state, the oxidation and the reduction reactions that occur within the muscle. Changes in muscle temperature, muscle tension, changes in metabolites, changes in circulating hormones. All of these can change during exercise and impact on the muscle. In recent years, there's been a lot of work on the various key enzymes and kinases, phosphatases, and various proteins that detect these signals. The calcium sensitive enzymes include calcium calmodulin dependent kinase, calcineurin. Energy status is monitored by the AMP activated protein kinase. There are mitogen activated protein kinases that seem to play a role in growth adaptations, and an important enzyme involved in protein synthesis known as mTOR. And certainly with the advances in molecular biology and cell biology, we understand a lot more about these molecular responses. And so how do these signaling pathways impact ultimately on gene expression, messenger RNA expression, protein expression? Various transcription factors, MEF2, GLUT4 enhancer factor, PGC1 alpha, which is thought to play an important role in mitochondrial biogenesis, are all transcription factors that have been implicated in the adaptive response of muscles to exercise. And more recently, interest in small non-coding pieces of RNA called microRNAs, which have been shown to influence the expression and transcription of genes and the expression of proteins. And ultimately, all of these changes are coordinated so that there's a change in either the expression and or the activity of various proteins which are fundamental for muscle performance and affect ultimately the functional properties of that muscle. So here in graphical form and nicely summarized, we can see various signals that are acting on a contracting muscle, either in an endurance type, prolonged exercise, high intensity exercise, but with dynamic exercise, through to more resistance type exercise, where the adaptations perhaps are more related to muscle mass or what we refer to as hypertrophy. And you can see all of the signals here, the circulating factors in the bloodstream, the metabolites that we're going to talk about in the fuels lectures, and how these can impact on various signaling kinases to either increase mitochondria or to increase muscle mass. If we focus on some of the adaptations to endurance type exercise, these are largely focused on increasing the oxidative capacity of the muscle. The mitochondria, who are the oxidative powerhouses of the muscle cell, they increase in their total volume in response to exercise, and that has important metabolic consequences. We see large increases in mitochondrial density and oxidative enzymes in response to endurance type exercise. We also see an increase in capillary density. You can see in this slide here the capillaries that circle muscle fibers, and there are more of them after training, and that facilitates the delivery of oxygen to the muscle and needs to occur in parallel with the increases in an oxidative capacity within the muscle. There's an increase in the glucose transport protein GLUT4, and that helps facilitate the storage of glycogen. There's an increase in the sodium-potassium ATPase activity, and this is an enzyme that's critically involved in the activation of the muscle, in limiting the loss of potassium from contracting muscle. And question mark on fibre type changes. I mentioned in the previous lecture, it's been ongoing discussion about whether prolonged endurance type exercise can induce fibre type changes. And there are some studies suggesting that that can occur. And certainly in animal studies with chronic long-term electrical stimulation, you see quite profound transformation of the characteristics of skeletal muscles. In terms of the functional consequences, we see reduced reliance on carbohydrate, reduced lactate production, and increased fat oxidation during exercise after endurance type training. And we'll talk more about this in the next module on fuels for exercise. From a health perspective, endurance training also improves the action of insulin, and that's thought to be very important in minimising the risk and indeed managing metabolic diseases such as type 2 diabetes. In terms of specific adaptations, let me give you just one example, and my apologies for being a bit parochial, but this is one of our research interests here at the University of Melbourne. How does endurance training impact on the expression of the GLUT4 transport protein? And here's one of our early studies where we examined cross-sectionally the differences between untrained and trained subjects in terms of their oxidative capacity as measured by the maximal activity of an enzyme in the mitochondria called citrate synthase, and the glucose transport protein GLUT4. And you can see that the trained subjects had higher muscle oxidative capacity and higher levels of GLUT4 compared with the untrained subjects. When we asked these trained subjects to stop their regular training for 10 days and report back to the lab, you can see that there was a reduction in their oxidative capacity and a reduction in their GLUT4 expression. So cessation of training reduced or diminished some of these adaptive responses. You'll note that they didn't fall all the way down to untrained levels, whether that would take a longer period of detraining or whether these athletes have some genetic characteristics which endow them with slightly higher muscle oxidative capacity or GLUT4, again, is one of those questions we may ask later in the course. If we look at the factors that regulate GLUT4 expression, and this seems like a complex slide, but it really reprises the theme in the very first slide about signals, enzymes that detect those signals, and functional consequences. So in the case of GLUT4 expression, there are two main signals that are thought to be important. One is the increase in calcium, which activates the calcium calmodulin dependent protein kinase, and the other are changes in the high energy metabolites, the change in the energy status of the muscle cell, which activates the AMP activated protein kinase. Both of these enzymes can target various transcription factors which bind to the GLUT4 gene, and ultimately changes in their localization and in their activity increase GLUT4 transcription so that you see increased GLUT4 mRNA after exercise in skeletal muscle. And ultimately, this increase in GLUT4 mRNA is translated into an increased GLUT4 protein. This increased GLUT4 protein means that you're better able to take up glucose in response to insulin and in response to contractions. In our next module, we'll talk about glucose uptake during exercise and the importance of GLUT4 for that process. An interesting observation that's been made for a number of years is that intensity can often be a very powerful stimulus. And here's a recent study comparing two types of exercise training designed to increase the functional capacity of muscle. And they measured the oxidative capacity again using the enzyme, the activity of the enzyme citrate synthase. And there were two types of exercise training employed. One was very high intensity exercise that you can see here, involved repeated 30-second sprints, 3 per week over about a 6-week period. The other was a more traditional endurance type training program, 40 to 60 minutes of cycling 5 times per week. You can see here over the entire training period, the high intensity exercise for a total of about 10 minutes, but at very high intensity, and the endurance training group for a total of 4.5 hours. And yet when you looked at the increases in citrate synthase, they were somewhat similar. There was no statistically significant difference between the increase. Whether that you would see changes in the long term remains to be seen, but it makes the point that intensity can be a powerful stimulus. And in time when people are increasingly time poor, there has been some interest in using these high intensity training programs in health and disease to try and promote these muscle adaptations. The other important adaptation that can occur in muscle is an increase in its size. And one of the hallmark adaptations to prolonged resistance type exercise, weightlifting type exercise, is an increase in muscle mass. You can see in this slide, relatively short exercise period, quite a large increase in the maximal voluntary contraction, the force generating capacity of the muscle, with relatively modest changes in the muscle cross-sectional area, a marker of the size of the muscle. So if you measure the EMG, or the electrical activity, you can see that much of the early increase in strength, functional strength, following resistance training is due to neurological adaptation. But over time, you do see increases in muscle mass. Conversely, if a muscle is subject to disuse because of injury, sickness, subjects are confined to bed, they have a cast, a range of reasons that might mean they're unable to fully activate their muscles on a regular basis, you can see that the myofibrillar protein synthesis rate decreases quite quickly. And in time, this results in a reduction in the muscle cross-sectional surface area. Again, there's been a lot of interest in the regulation of muscle mass because of the importance of muscle mass in health and disease. For athletes, it's important because it's the force-generating tissue. In normal, healthy individuals, an adequate muscle mass is important because of the role that skeletal muscle has in supporting movement, supporting body functions, and supporting metabolism. And one of the consequences of aging and a number of disease states is a loss of muscle mass, and that can impact on functional capacity and the quality of life. So there's been a lot of interest in the regulation of muscle mass. In terms of building muscle, an important enzyme appears to be mTOR, and this has an important role in protein synthesis. It's influenced by a number of growth factors acting through insulin-like pathways. On the other side, factors which influence the degradation of muscle or muscle atrophy, there's been a lot of interest in the so-called ubiquitin ligases, which are involved in breaking down muscle and how that might be regulated. In relation to exercise effects on one of those signalling proteins, mTOR, what we see is that exercise activates that enzyme, at least as measured by its phosphorylation status, which is the usual way that we measure the activation of these proteins. If you look at protein synthesis, and here is the measure, the fractional synthetic rate for protein, during exercise, the protein synthesis rate actually goes down. And given that exercise is an energy demanding process and protein synthesis requires energy, it makes sense to perhaps turn that off while you're exercising. But you can see after exercise an increase in protein synthesis. And so with regular bouts of exercise, regular activation of mTOR and various other protein synthetic pathways, you get an increase, a cumulative increase in protein, myofibrillar protein, the muscle gets bigger and the muscle gets stronger. And finally, one interesting aspect of muscle adaptation to exercise that's really been studied in great detail in the last decade or so, and that's the observation that contracting muscles in fact release biologically active substances, and these have been termed myokines. And don't worry about the detail in this slide in terms of specific myokines, but you can see a number of them are released, and they have effects on adipose tissue, on the liver, perhaps on the pancreas, on the bone, on blood vessels. And this raises the interesting possibility and likelihood that these biologically active compounds that are released from contracting muscle have effects on tissues distant from the muscle and may account, at least partly, for the systemic benefits of regular muscular exercise in promoting health and well-being.